everyone. Have you ever felt too ill to get out of bed? And you might think of a hangover right now. But no. We're speaking about a serious disease. Imagine that you're too weak to get out of bed every single day. And that there are no effective treatments. This is the case for many patients with an autoimmune disease. A disease in which your own immune system starts to attack your own cells. My name is Jacob, and I'm here together with Jolien, and we are from iGEM TU-Eindhoven, and for the next 15 minutes, we'll explain to you the solution to effectively treat autoimmune diseases. Worldwide, 400 million people suffer from autoimmune diseases. And we were shocked that for most of these diseases that affect so many people, there are still no effective treatments. And that is why we wanted to make an impact by coming up with a new, innovative therapy. But since there are over 80 different autoimmune diseases, where do we start? So initially we had 2 autoimmune diseases in mind. But then, when we dived deeper into the literature and when we reached out to experts, such as university medical centers and hospitals, to find the autoimmune disease with a high need for a new treatment, they urged us to switch to the disease called ANCA-associated vasculitis, or in short, AAV. First, because of the severity of the disease, and secondly, because of the lack of effective treatments. And at this moment, worldwide, there are 1 million people suffering from AAV, which is a systemic autoimmune disease that is characterized by inflammation of the small blood vessels. And this inflammation leads to blood vessel necrosis and therefore lack of insufficient oxygen supply towards the connected organs. And eventually this leads to decrease in tissue functioning or even tissue death. And to understand the requirements for a new therapy, we engage with multiple patients. Take, for example, Louis, who is diagnosed with AAV since 2007. And in the meantime, due to this disease, Louis has lost the top of his fingers, he's had 2.5 years of kidney dialysis, and he's experienced countless numbers of hospitalizations. But do you know what the most intriguing thing was that he said to us? I did not dare to sleep at night when I was alone, because I was convinced I would not wake up anymore. And the treatments that he received are immunosuppressive therapies that non-specifically suppress the entire immune system. And the patients we interviewed, but also the hospitals and literature state that these current therapies are ineffective for multiple reasons. Because, first of all, a large part of the patients do not respond to them. Half of the patients experiences relapses within 5 years. The current treatments are inherent to severe side effects, and because of the intensive treatment procedure, hospitals experience a very high workload and high costs. So, based on the conversations with the experts, we have now learned that there is a significant need for a more specific treatment against AAV that, first of all, prevents relapses, secondly, reduces side effects, and lastly, also reduces the workload for the hospitals. And that is why we, as iGEM TU-Eindhoven, with nearly 30 different stakeholders, have co-created Impact, a modular, personalized autoimmune cell therapy. So Jolien, how does Impact work?
Well, as Jacob already mentioned, Impact is a cell therapy. A therapy where engineered immune cells are administered to the patient. And you might already have heard of a cell therapy before. There's a quite well-known example already on the market. The CAR-T therapy to cure various cancers. And our therapy is similar. The treatment of Impact works as follows. Harvested immune cells from the patient get genetically engineered by incorporating the genes of a synthetic receptor. Those cells get expanded and get injected back into the patient. In the body, the engineered cells detect the pathogenic autoantibodies of the disease. and binding of these antibodies to the receptor on the cell will activate the system and will result in the production of an anti-inflammatory cytokine, interleukin-10, also referred as IL-10. And after a pivot in our concept, engagement with stakeholders resulted in this final design. Hospitals, university medical centers, and the pharmaceutical company stressed the potential of IL-10 in treating autoimmune diseases. In addition, IL-10 is already proven to be effective in multiple studies. The technology of IMPACT is based on the Generalized Extracellular Molecule Sensor Platform, or in short, GEMS. This is a modified human receptor that dimerizes upon binding of a molecule, and it induces a signaling pathway. This modular synthetic GEMS receptor allows the coupling of an extracellular input to an intracellular signaling pathway. And in our case, binding of the antibodies to the receptor activates the receptor and induces the signaling pathway. Eventually, it will result in the secretion of IL-10. And the activity of IMPACT is dependent on the antibody concentration. It is thereby adjusted to the disease activity, making the treatment personalized. This reduces the side effects and it prevents relapses of the disease. Well, to validate our design, we tested the proof of concept in the lab. We supported it with a kinetic model and we validated it with experts. Well, so we first started by building upon the original work, and we started by checking the transfection and the activation of the original GEMS receptor. To realize this, we transfected plasmids in mammalian cells, including a plasmid encoding for the original GEMS receptor and a plasmid encoding for the reporter protein. We successfully showed receptor activation upon binding of small molecules, and higher ligand concentrations resulted in more activation of the receptor.
So Jolien, how did you then prove our concept? How did we validate our proof of concept?
Well, for our proof of concept, we divided the system into two parts. On the one hand, our goal was to change the output of the receptor from the expression of a reporter protein into the expression of a therapeutic protein, IL-10. And our second goal was to change the input of the system so that antibodies could activate the receptor. First, to test whether our engineered cells were able to produce IL-10, we modified the plasmid encoding for the reporter protein so that it encodes for IL-10 using cloning strategies. And we again transfected plasmids in the mammalian cells, but now plasmid encoding for IL-10 instead of the reporter protein. And to quantify the IL-10 expression, we made use of an ELISA assay. Binding of the original ligand to the receptor resulted in a 34-fold increase in IL-10 concentration. So we successfully designed a GEMS receptor to be a therapeutic instead of only a diagnostic. Well, the next step was to achieve receptor activation by binding of antibodies. And as this is never done before, we first investigated the binding of antibodies to the receptor. And we started by changing the extracellular affinity domains of the receptor so that antibodies could bind. Then we executed flow cytometry experiments, and therefore we transfected the receptor constructs in mammalian cells. And those cells were stained with labeled antibodies. An antibody binding of around 20% was seen. And these results are promising, as it's the first time antibody binding to a GEMS receptor is shown. So, the next step was to check the activation of the receptor by antibodies. However, no significant activation was seen yet. To improve the binding of antibodies to the receptor, we started optimizing the distances between the affinity domains of the receptor, so that antibodies could bind well, and in turn could activate the system. In conclusion, we have successfully designed a GEMS receptor to be a therapeutic instead of only a diagnostic. In addition, we are optimizing the binding of antibodies to the receptor to enable antibodies as an input for the GEMS receptor. And throughout this process, we received feedback from multiple stakeholders, also on an aspect that might be critical for us. And we covered it, right, Jacob?
Indeed. The National Institute for Public Health and the Environment in the Netherlands explained that the immune system is very tightly regulated, and an overproduction of IL-10 could lead to dysregulation of the immune system. And they still encourage us to use IL-10, but they also encourage us to find a way to control the IL-10 production. And that is why we have built a kinetic model in MATLAB based on ordinary differential equations of our cell-based system. And with this model, we have performed a multiple parameter sensitivity analysis to find sensitive parameters that can be tweaked in the lab to get the desired IL-10 production. And the results of this model show that the IL-10 production is highly dependent of the binding affinity between the antibody and the receptor. So for future work, by changing this binding affinity, we can optimize this process to get the desired IL-10 production of our engineered cells. So during the course of this presentation, we have now seen how the human practices work really shape the core and the direction of our project. But the experts also helped us to make the translation of our project into the real market. Small medtech startups, but also large pharmaceutical companies, therefore helped us by creating a business model to start a successful new venture after the Grand Jamboree.
And of course, we want to enter the market in a responsible way.
Indeed. And that is why, with experts on cell therapies, but also experts on the risks of synthetic biology, we have confirmed the safety of our engineered cells for the patient and also for the environment. And for this, we used the risk assessment of the already FDA-approved CAR T cell therapy, because, as Jolien explained, these have high similarity to our therapy. And moreover, next to building a model, we also proposed a kill switch to overcome the potential challenge of IL-10 overproduction. And lastly, together with experts on biosecurity, we proposed a strategy to prevent possible dual use of our technology. So next what we did is we identified all the steps that are required to reach our final end users, who are the hospitals and the patients. And during this process, we paid special attention to the step of market authorization and reimbursement of our cell therapy in the health insurance. Because these two steps are largely different for cell therapies than for other medicine. And to find out whether our end users, the patients, are willing to receive our cell therapy, we also, together with iGEM Boco Vienna, have performed a survey among the general public to find out what they think of medicine based on synthetic biology. And the results showed that a large part of the society is still not yet familiar with synthetic biology. And as a consequence, many people, independent of age, are afraid of the use of synthetic biology in medicine. And that is why we have educated different age groups of the society, and we reached out to them by means of publications, presentations, SynBio events, podcasts, social media, and many, many more, which can be viewed on our wiki. So, Jolien, what can we now conclude based on our story?
Well, in conclusion, we have designed a well-validated, modular and personalized cell therapy to overcome the unmet need in current treatments against AAV.
And how do we overcome this unmet need?
Well, impact is unique since, first of all, it prevents relapses of the disease, as the engineered cells immediately activate upon increased antibody concentration. Secondly, it leads to fewer burdensome side effects. And lastly, it reduces workload and cost of the hospitals by preventing the relapses of the disease, further health damage, and the associated hospitalization. And there's even more. The modularity of our innovative platform technology offers the potential for our treatment to be applicable for multiple autoimmune diseases. And with 400 million people suffering from autoimmune diseases, our therapy can eventually help many people and thereby reduce healthcare workloads. This modularity is shown in our part collection that can be found on our wiki. And our best new composite part and the associated best new basic part are validated to work and are now available for the iGEM community. We would also like to take this opportunity to thank all our sponsors and everyone who helped us throughout this process. But our journey is not yet finished.
It is just the beginning.
So do you want to join our mission to innovate autoimmune disease therapies? Please come to us after this presentation to see how you can contribute. Thank you.